This presentation is on a couple of different aspects related to neuroplasticity. It's a senior seminar course I teach here at Virginia Tech. This is related to a lecture on adult neurogenesis in the olfactory system. It's mostly to make up for the fact that we ran out of time on a recent lecture I did in person, and I wanted to provide a little bit more explanation and cover two research articles that I feel like exemplify some of the interesting research in this topic, and it's probably of interest to a pretty wide audience. So to get everyone up on speed, I'm going to give a very, very brief overview of the topic, which is adult neurogenesis. So in general, it is thought that, or at least for many years, it was thought that the brain is mostly fully populated with all the neurons it's going to have at a very young age. And evidence indicates that by around 18 months of age, almost all the neurons that will be born in the adult human brain are born. There are some exceptions to this, though. In humans, there's reasonably good evidence, there's some debate about this, but reasonably good evidence that there is ongoing adult neurogenesis happening within the hippocampus. Now there's less evidence, in fact, probably very little evidence, that there's neurogenesis happening in other areas, although there's debate about that as well. In rodent brains, we know that there's also neurogenesis happening in the hippocampus, and this is adults that we're talking about, right? But also in rodents, we know that there's substantial neurogenesis happening in the olfactory system. And that's what we're talking about today. There's not good evidence at all that this happens in humans, at least neurogenesis into the olfactory bulb. As far as the olfactory epithelium goes, that's slightly different. So to give just a brief overview of the olfactory system in mice, this is the nair, where we breathe in, or the mouse would breathe in air. And the mouse, well, all mammals have two olfactory systems. One is called the main olfactory system, that's in blue, and in pink is the accessory olfactory system. Now pheromones are, the accessory olfactory system is specifically there to recognize pheromones, these chemicals that are made by conspecifics in order to provide sort of a communication or stimulatory reaction in the animal. Now the main olfactory system also senses pheromones. It does play a role in that, but this is the area where it senses all the chemicals that are in the air, and we refer to those as odorants. So the odorants will come in, they'll act on the receptors and the main olfactory epithelium, which in all mammals, as far as we know, there is ongoing neurogenesis there of the olfactory receptor neurons. This is including in humans. And then from there, that information is passed on to the main olfactory bulb, where we see substantial neurogenesis happening in rodents. From there, odorant information is passed on to other areas of the brain, and that's beyond the scope of what I'm talking about today. So what is it, to give a very brief overview of what this adult neurogenesis looks like in the olfactory bulb, I've got this nice figure here. So the neurogenesis in the olfactory system it's a great representation of the four basic principles of neurogenesis. There are four major stages that characterize neurogenesis, and these are the things that you would have to have, more or less, in order to have ongoing neurogenesis in adults. So you have to have proliferation, so that is the division of new cells. Now those cells, generally, those cells are not born in the area where the neurons are needed. They have to migrate to those areas where it's needed. And then they need to differentiate, and then they have to be integrated into existing neural circuits. So it's those four basic steps: proliferation, migration, differentiation, and incorporation. In the olfactory system, the cells that are born, that will end up being adult-born neurons, are born in the subventricular zone against the lateral ventricle. So this is representing where those cell divisions are occurring, and that's pretty far deep in the brain, far meaning in the caudal axis, so towards the back. This is the back of the brain, this is the front of the brain. These cells, they're born here, but the olfactory bulb is over here, so they have to migrate down to the olfactory bulb, and they migrate through what's called the rostral migratory stream, RMS. And along the migration pathway. Those cells are undergoing the process of differentiation, which largely is completed by the time that they move into the olfactory bulb. And then from there, those very young neurons need to be fully integrated into existing neural circuits. And so that's the integration part. So then they have to grow dendrites and axons and connect up to other neurons. Neurogenesis in the olfactory bulb is restricted to just a couple of particular types. Now there's a bunch of different neuron types. There are the receptor neurons in the olfactory epithelium. That's outside of the olfactory bulb. And then those neurons will send their projections down to the olfactory bulb, which, the olfactory bulb is actually quite beautiful. This is a cross section of the olfactory bulb. And the outermost layer of the olfactory bulb, you're going to see these like bubbles that are within the tissue. So this is like mouse tissue right here. And this blue stain is labeling all the cells. So the areas where you don't see any blue or see very little blue, those are going to be mostly filled with axons and dendrites. And so these are known in the olfactory bulb, they're known as glomeruli. And each glomerulus will be processing odorant information from a specific odorant receptor. So that's where you start to get organization of odorant information in an individual glomerulus. Then it's those neurons, they're synapsing onto the dendrites of cells that are known as mitral cells or tufted cells. And the cell bodies of those are located here. And they have a unique shape that looks kind of like a miter, like a big fancy hammer from, the Knights of the Round Table type times. So those cells then send their information down to a whole bunch of downstream targets into the cortex and other areas of the brain. Another important population within the olfactory bulb is the granule cell layer. So this is filled with these tiny little neurons called granule cells. These are inhibitory neurons, and they actually are getting information from upstream down up in the brain, and they are modulating the activity coming into the olfactory bulb by synapsing onto the mitral cells and also synapsing onto themselves. You can see one of them right here. And these are inhibitory neurons, so they're regulating the overall activity coming out of the olfactory system. So there's a lot of programming that can happen within just the olfactory bulb because of this feed-forward network that goes into the granule cells. Now, one thing that we know is that if you inject animals with BRDU in order to label new cells that are dividing in the animal, including in the brain, and then you wait 40 days after you inject those animals with BRDU, and you look in the olfactory bulb. So it's the same sort of profile that we're looking at right here, except it's turned 90 degrees. So this is the granule cell layer here. And they're using immunofluorescence, so we're seeing the bright cells. Those bright cells are immunolabeled BRDU positive cells. And we see bright cells here in the granule cell layer. So these are BRDU positive cells in the granule cell layer and BRDU positive cells in the glomerular layer. And further work has shown that the new neurons born in the, that make their way to the glomerular layer are known as periglomerular cells. So that's one of these guys right here. So those are the ones that are dividing. And then there's a ton of neurogenesis happening that for new neurons that end up being granule cells. So there's quite a bit of new neurons that are added to this existing circuitry. The question is, like, what kind of functional role might this play? So that's the part that I'm going to be talking about today. So I wanted to discuss two papers, just a few figures from two of these papers. So this is a paper published from 2007, so quite a bit time ago. It's from Gloria Mack et al. You can see the authors listed here. The title of the paper is Male Pheromone Stimulated Neurogenesis in the Adult Female Brain, Possible Role in Mating Behavior. Now, there's a lot of background information that's being left out for those who are just seeing this video on their own. You'll have to take my neuroplasticity course if you want to get the full story. For my neuroplasticity students, you should have seen the lecture that I gave in class, and then this is where we're catching up. So the first thing I wanted to show is data from this figure. It shows that females spend more time sniffing dominant males when exposed to dominant male pheromones. So this is a strictly behavioral aspect of what they're showing here. So we can take mice and do certain behavioral things. Mice tend to organize in a dominant-subordinate relationship as far as males are concerned. So male mice like to fight when they're put together. They're not very friendly with each other, and they generally will quickly establish dominance or subordinates. And so if you do this with enough mice, you can figure out which ones are subordinate and which ones are dominant. That can change. There's a whole bunch of work around that. But they've got established dominant male mice and established subordinate male mice. Now, what they did prior to doing the behavioral testing, they exposed the females to the bedding taken from either a dominant male or a subordinate male. And so the subordinate female exposed would be the FM sub, so that's these ladies here. And then the ones that were given bedding from dominant male, so they weren't exposed to the males, just the bedding, they're shown here. Now, and then after 7 days, they put them in this kind of testing cage where they look to see where females that are exposed to either the subordinate male pheromones or dominant male pheromones, how much time do they spend sniffing the different males? So they can go from one side to the other side. It's kind of like speed dating for mice, except you had to smell their pee for a few days, which is kind of gross. So for the subordinate females, so they're exposed to the subordinate female pheromone. There really isn't any difference. They don't spend any additional time sniffing the dominant or the subordinate. Subordinate is black, dominant is white. The white bars. But for those females that were exposed to dominant male pheromones for 7 days, they spend a significantly greater amount of time sniffing the dominant male. Interesting. So just that pre-exposure to just the pheromone changes the way those females react in the once they're put into the cage and allowed to choose between the two males. Another result from the study is looking at the effects of exposure to male pheromone on proliferation in the subventricular zone. So this is looking at the proliferation part. So females that are paired with another female is shown here. These are brain sections stained with BRDU. So they did injections of BRDU and then killed them not 40 days later, but I think in this study it was either a day or 2 hours later, very quickly, just so that you're labeling the proliferating cells. They wouldn't have time to make their all the way down to the olfactory bulb. And this is females paired with a female, and these are females paired with a male. You can see the females paired with a male have a significantly greater number of proliferating cells in the subventricular zone. So females just being around another male have more BRDU positive cells in the subventricular zone compared to females with a female or females just by themselves. So there's increasing neurogenesis. Another result that they have in here is that they had females that were either paired with a male that was castrated, so he had no testes, or a male that had sham surgery. So they went through the surgery procedure, but they didn't take out the testes. That's to account for like any damage the surgery might do. And so those females that are exposed to the sham males, they have elevated neurogenesis, but the ones that are castrated do not. So this indicates that, okay, there must be something about the maleness, some kind of male hormonally driven pheromone is likely being released. One way to test this is to treat the females with zinc sulfate. And so in this case, what they do is they squirt a little bit of zinc sulfate up into the nose, and that kills cells within the olfactory epithelium. So the females can't really smell very well. So females that got that procedure, they have reduced neurogenesis compared to the ones that got saline. So they just had saline squirted in their nose. So males that are intact are producing something that's making females have elevated neurogenesis, and females have to be able to smell it. And even more interesting is that females that are exposed to dominant males, so they've been established as dominant, they have this increased neurogenesis, whereas females that are exposed to the pheromone of subordinate males do not. So there's something about dominant males that also makes this happen. So a bunch of really cool results all in one little figure here. So they wanted to test, is this neurogenesis important? So one way, and so for this you have to do certain experiments. You gotta make some sort of manipulation of the system. What they did here is that they infused into the brain a mitotic blocker called AraC. So the only thing we really need to know is that this blocks mitosis, cell division, so that's going to reduce neurogenesis. And just to see if this is indeed the case, here's just the example of what this looks like. So you have females that were infused with saline, or females that were infused with AraC. So this, again, you have to have a proper control to control for the surgery procedure. That's why you have saline. And this looks just like females that weren't infused with anything, right? The ones that have exposed to dominant male pheromones, they have increased neurogenesis compared to the subordinates. But in both groups, you can knock down neurogenesis to virtually nothing. This is just basically showing that the AraC is completely reversing this phenomenon of neurogenesis happening in the olfactory bulb. So AraC seems to work. We can block neurogenesis. So females that are treated with AraC no longer prefer dominant males. So here, in this case, we have females that were exposed to subordinate conditions, and then they were given and then also infused with saline or AraC, and they were given the choice to spend time with either a subordinate male or a dominant male. And remember, so this is kind of like the control experiment from before, where you wouldn't, if we're replicating what we saw before, the first panel in this study, we wouldn't expect to see a difference in duration of sniffing, and indeed that's the case. And a female that's exposed to subordinate male pheromones that also had neurogenesis blocked, no difference. Okay, this is where it gets interesting. So females that have that are exposed to the male, the pheromones of dominant males that and then we look to see how much time they spend sniffing. For those females that were infused with saline, they spend a significantly greater amount of time sniffing the dominant male. So this is what we would expect. This is replicating the panel from that first study, where they showed that those females exposed to dominant male pheromones for 7 days now want to spend time with dominant males. But if those females were infused with AraC, which blocks neurogenesis, now these females have no preference for the smelling. So this is showing that this neurogenesis is necessary in order for the females that are exposed to male pheromone to have this preference for dominant males. So a really cool study. There's a lot more in the study, other interesting panels. If you want to learn more about it, I encourage you to check it out. Right here, there's the reference. The second paper we're going to talk about is entitled Activation of Adult-Born Neurons Facilitates Learning and Memory. So this paper, just like the previous paper, was published in Nature Neuroscience. This one was published about 5 years later, so this is 2012. As far as neuroscience standards go, this is now a relatively old paper. There's a lot of interesting results in this paper, however. This is by Alonso et al. Here's all the authors. This study uses a method referred to as optogenetics. And so this is, optogenetics refers to that kind of field that uses a combination of techniques of optics, meaning like using light, and genetics to control the and or monitor the activities of individual neurons in living tissue. And that's at least referring to the neural component of optogenetics. This study is using one of the more commonly used optogenetic tools, which is called channel rhodopsin. So channel rhodopsin is it's a light-sensitive channel. It's taken, I believe, channel rhodopsin is taken from algae or bacteria. I don't remember exactly. It's from some other animal, but it's been modified and changed and optimized for use in all kinds of animals. And so we can transfect tissues in animals to express channel rhodopsin, including neurons in the brain, so that we can now control neural activity by just shining a light on those neurons. So for channel rhodopsin, you would shine a blue light onto those cells, and then that would open up the channel rhodopsin that would allow sodium to flow in, and so with sodium flowing in, you're going to have depolarization, which is going to cause an action potential. You can do brief pulses to have like a firing rate, and it's a very effective tool for controlling overall neural activity. It's almost science fiction type stuff, but it's a super exciting tool. We even use it in my lab. So optogenetics can be used to activate newborn neurons. So this is really a very cool technique. In this study, what they did is they injected a lentiviral vector that encodes the channel rhodopsin. It also has attached to it a fluorescent protein so they can see those cells that are labeled. In this case, they're using yellow fluorescent protein, or YFP. So they're injecting this into the rostral migratory stream where there's some cell division happening within the rostral migratory stream, right very close to the subventricular zone. This lentivirus then gets taken up by those cells as they're moving through and dividing. So the only cells that are going to be labeled are going to be those cells that are young and dividing that take up the lentiviral vector. Now, if you do this way back here and then you look into the olfactory bulb, and after waiting a little while, you can look and by a little while I mean weeks, maybe even months, you will see yellow fluorescently stained cells. In this case, they're just showing green because green is a little easier to see than yellow. They're just changing the color a little bit, but these would have been yellow. And that would mean that these cells, they look like neurons, they have long processes. This is in the granule layer, this is the periglomerular cells, so they got both kinds labeled with the green, or yellow in this case. They're labeled, right? This would also mean if they have the YFP, they also are expressing channel rhodopsin, which means if they shine light on them, they will get them to glow. So what they did is then they made a hole at the top of the nose, going through, or like above the olfactory bulb, and placed an LED on the top of the brain. So fortunately, they were able then to emit this blue LED that's controlled to a computer where they can just stimulate the cells with brief pulses of light. And then this is showing the activity of these channel rhodopsin YFP neurons, where you get action potentials, you get changes in current. This is entirely expected and very precisely controlled by the brief pulses of light. 200 milliseconds is the time course here, so you can see like really, really tight control. So now you can control activity very precisely of these newborn neurons. So the existing adult-born neurons, the existing ones that were already there before we did the labeling, they're not labeled. We're only controlling newborn neurons with this procedure because we're labeling them way back here where they were being born with the virus. So very cool way of setting this up. The channel rhodopsin, when activated, seems to have an enhance in performance. And so this is what we're going to walk through here with these data as they showed it here. In this case, what we're seeing is that the mouse, when they're presented with either the rewarding odor or the odor in which they're supposed to avoid, they have to make the right choice, and then they will get a reward. So if they reject, they will get a reward, but they have to move away. If they are presented with the correct odor that they have to focus on, and they come over here and hit the button, they also get a reward. So they got to make the right choice. They can make a false choice with those two. And as they go through them, those are called blocks, and so they're going up through the blocks. And what we see here is that they have 3 different light stimulation protocols that they're doing. So they're either stimulating the neurons at 40 hertz, so 40 times per second, or 10 hertz, which is just 10 times per second. So this is relatively slow, this is relatively fast. And they're either doing it at the very beginning when the odor is presented, or they're doing it later, delayed, after the odor is presented, and before they're supposed to make the choice. And that's for the 40 hertz phenomenon. For the 10 hertz, there's a delay. So what they show here, what we can see, those animals that are getting the 40 hertz phenomenon and are getting the channel rhodopsin, they have an enhanced, how to describe this? As they go through the number of blocks and they're getting challenged more and more, what we can see is that the chances of the mouse getting the percent correct is better if they're getting the channel rhodopsin stimulation at 40 hertz during the odor presentation phase. So by activating those adult-born neurons, remember these are adult-born brand new neurons, stimulating them with this overall stimulation to kind of make them a little bit more excited, it enhances the memory performance, the ability for the mouse to discriminate between these two odors and get them correct faster than under control conditions. You can see that they get up to about 80% correct after about 20 blocks, where it takes the controls about 30 blocks or so to get close to 80%. So it's a really interesting enhancement of performance. So this is looking at this data in a little bit more broken down detail. So under easy conditions, so making the odors actually very, very different from each other, the blocks to criterion, meaning like getting up to a certain percent where, all right, it seems like they've learned it. It doesn't actually make a difference for the channel rhodopsin versus the controls. So if it's really easy, channel rhodopsin isn't going to make it any better because it's already really easy for the controls to figure this out. But when you put it under difficult conditions, using odors that are tough to discriminate, when you have the channel rhodopsin under the paired condition where it's being presented with the odor, you can see the blocks to criterion is significantly better than the controls. So they learn it faster than the controls, the animals that don't get the stimulation. Under the delayed condition, you see that there's no difference. Now the animals, so when they're waiting a little bit after the odor and then they stimulate, that's not actually helping anything. It has to be during odor presentation to get the improvement in performance. And it appears that there's a certain sort of rate of stimulation that these neurons like, which is around 40 hertz, and because 10 hertz is insufficient to induce enhanced memory performance. And even further, they did a correlation analysis of their data. So when you do this kind of labeling procedure, you're not going to get the same exact number of neurons across all animals. You're going to have some animals that will have a lot of these channel rhodopsin YFP positive neurons, like around 600 or so, and you'll have some that will be less, around 400, so about 2/3 of what is maximal. And for those animals that have more channel rhodopsin positive neurons, they need fewer blocks to reach criterion, whereas if they have fewer channel rhodopsin positive neurons, they need more blocks to criterion because the stimulation is hitting fewer neurons here, right? Because there are only so many that were hit. Whereas if they have a lot more, they can do better. So enhanced performance is also just correlated with the number of cells. So a really cool study showing that just enhancing overall neural activity of these adult-born neurons during the presentation phase, when they would actually be used, and at a particular rate, 40 hertz, can enhance memory in these animals. So it shows that these adult-born neurons, they can confer a substantial benefit if they're being stimulated in a certain way. So that's it. Those are just two studies that I think show some interesting results on the significance of adult-born neurons in the olfactory system in rodents. There's a lot of others. I encourage you to look through the literature, find more for yourself. And yeah, there we go. Have a good day. Thanks for listening. Please like the video and subscribe to the channel if you'd like to see more content like this. Take care. Please like this video and subscribe to the channel if you'd like to see more content like this. Looking to make more videos like this on neuroscience, on interesting papers, and also on other science-related topics. So take care. Bye-bye.